Hello and welcome to my exam mini video on Francis Crick's adaptor hypothesis. Francis Crick once said, "Almost all aspects of life are engineered at the molecular level, and without understanding molecules, we can only have a very sketchy understanding of life itself." With Crick's quote and perspective in mind, let's explain further the adaptor hypothesis through the protein synthesis garden illustration. So first you should know what each aspect of our garden represents. The flower bed is the mRNA chain, and each flower is the codons that are made up of U, A, G, and Cs, which you see here by the different colored flowers. All the codons are read in sets of 3, so the butterflies are representing the adaptor, or drumroll, the tRNA. And just like there are different types of tRNA, there are also different types of butterflies. Then if you notice, hovering over the butterflies is the bumblebees that represent the amino acids the butterflies quote-unquote translate or produce from the flower mRNA. All the bumblebees are in a chain tied by their flying pattern that represents the polypeptide chain. Then lastly, we cannot forget about the cloud surrounding the butterfly tRNA, which is representing the ribosomes where this whole process takes place. So, the adaptor hypothesis. Crick had this idea that a molecule would match amino acids to codons from the mRNA sequence using an adapter. The adapter would make it possible for the amino acid to connect to the mRNA template no matter the codon sequence. It would adapt to receive any specific amino acid and interact with the mRNA, hence the name. The adapter would then be able to incorporate the amino acid into the growing polypeptide chain. The adapter could roll with the punches and be flexible yet consistent in producing the suitable amino acids needed. This was just a hypothesis that led to the later factual definition and molecule of tRNA. As we mentioned before, our tRNA butterflies are the link and key for the protein synthesis garden and all the protein synthesis happening in our bodies. So as we discussed, tRNA, the adapter molecule, takes mRNA codon sequences and links them to an amino acid specific to their codons. tRNA follows the universal genetic code and consistently adapting to match the same codon sets with amino acids always. However, sometimes the tRNA is prevented from being an adapter, and there are consequences because of that in our cells. An example of this can be seen in human mitochondria in the codon code UGA. It is supposed to signal a stop in the amino acid chain. However, the tRNA doesn't adapt and instead links TRP. You can see this represented in a more scientific genetic illustration to the right of the screen. When this happens, it causes negative health conditions in our organs, specifically in their growth and energy capability. This is important in having our organs like our heart and lungs. Thank you for watching.